Okay, welcome everybody to this edition of the ICANX Youth Talks. My name is Jonathan Rivnay. I'm a professor of biomedical engineering and material science at Northwestern University. My co-moderator today is Alexandra Rutz from Washington University in St. Louis. And we're pleased today to host three great talks, amazing scientists for these youth talks, really rising stars. We have Anna Maria Pappa from Khalifa University. We have Helen Tran from University of Toronto. And we have Sihong Wang from University of Chicago, talking about all aspects of bioelectronics and specifically how soft matter can be used to interface with biological systems. It's a real pleasure to introduce these three speakers. So our first speaker today will be Anna Maria Pappa from Khalifa University. Anna Maria, I'll let you share your screen and I will introduce you. Okay, so Anna Maria joins us from Khalifa University, where she's an assistant professor in the Department of Biomedical Engineering. Previously, she did a PhD in the École des Mines de Saint-Étienne and a postdoc at Cambridge University in the UK. Amongst her numerous awards, she's received a L'Oréal UNESCO Women in Science Award, as well as a 35 under 35 in Europe for the MIT Technology Review. Anna Maria, we're looking forward to your talk.
Thank you so much, Jonathan, for this introduction. I'm really happy to be here actually among in this great audience and to be talking for my research among these great scientists, Helen, Tran, and Sihong Wang. I guess I will kick off this seminar series with my presentation on cell membranes on chip. So I would like to start with what is actually a cell membrane for a cell. So it is actually the gatekeeper of the cell. It regulates the traffic into and out of the cells. And this happens via specialized membrane proteins that can be, for example, ion channels. Its structure is like a lipid, as you can see, enveloped lipid bilayer, and you can see membranes spanning between and around the lipids. And those proteins, cell membrane proteins, are actually responsible for all the functions of the cell membrane and protect it. So no one can argue it's very significant to be able to study the cell membrane, but why it's not as much studied as the cells are? So the answer to that is that it's very difficult to study cell membranes because cell membranes are really dynamic. And by dynamic, I mean lipids and proteins, they constantly move and they shift and rearrange themselves as per the cell's needs. And this dynamism really makes their study exceedingly difficult. Membranes are also intrinsically disordered, which means we cannot study them with techniques, for example, like crystallography. And so what we need is to actually find a way to provide to the membranes the necessary stability, a support, if you like, so that we can experimentally study them. But what is the key challenge in this is that we need to maintain their fluidity so that their dynamism and these dynamic changes that happen over time during the life of the cell, they can continue happening because the membrane will be mobile and fluid. So before I go on to how we do that in my lab, I would like to talk to you about what I like to say the future hope. So why do we study? Why it's important to study in vitro? Of course, you are, I guess, all familiar with the fact that we can take our very own cells and reprogram them, differentiate them, render them to whatever we need to study, and then study therapies, drugs within our very own cells, but without the need to put those drugs in our body. So we can study the drugs and their effect, whether they are safe or not, whether they do the job they promise to do on our very own cells outside our body. And no one would argue that in terms of bioengineering and biology, this is actually, biology has evolved, and we are at that stage. What we do lack, though, is the technologies to interface our cells, our in vitro, let's say, biological systems, in order to allow for dynamic functional assessment. In other words, we lack the technology to really read and understand those biological models that we can reliably make. So, having said that, in my lab and other many labs, we focus on bridging the gap between biology and technology. And this through both design, device, fabrication, integration, and of course the right data analysis. And of course, biology in vitro differs in terms of length, scale, and complexity significantly. We can go from one protein to a cell membrane, to a single cell, a cell tissue, even 3D cell cultures, or even organoids. So those are biological systems of different complexity that we can interface with devices, with technology, if you like, and study them outside our body. Now, what is really important in that is that given the different length and complexity levels of those systems, we need to co-design. Along with the biological model, we need to co-design the technologies, the devices, so that they can accommodate biology and sensitively read the signals from biology. And as I said before, I'm very interested on the cell membrane. But how do we reconstitute? How do we reliably develop models of the cell membrane to study them? So a very simple model that I can show you is what we call a lipid-only, or you might see it as a non-authentic or artificial cell membrane, which consists only of lipids organized in a bilayer format, and they can be either on, like in this vesicle format that you can see, here, or they can be organized in supported lipid bilayers on top of solid supports. Most of the times, this is glass or mica. In order to add the proteins we're interested to study, we can reconstitute what we say, the protein of interest, and then perform studies. However, this is not the real thing, right? And this is the latest, actually, approach that many scientists, including us, is using, which is getting natural vesicles. Those vesicles actually come from the actual cell, as you can see here. Those are plasma membrane vesicles, and we can use them and make them in a bilayer format. I will explain you later how. And now we have the actual membrane of the cell without having to deal with the whole cell. So we are more sensitive on what happens at the membrane, at the first point of contact of the cell membrane. So how do we do that? This was a technique developed by the Daniel group in Cornell. So you get the cell, whatever cell that is, and then you can do some synthetic engineering. You can overexpress, for example, a certain protein of interest or not. And then you can add some chemicals which would induce what we call blebbing. So blebs are blister-like protrusions in the cell membrane. And when we add this chemical solution, we sort of amplify this process, and the cells actually undergoing a pre-apoptotic stage. They start producing a lot of those vesicles, and then we can collect them on a surface, the surface we're interested in. And then those vesicles will fuse. It's called vesicle fusion, like in a parachute mechanism, and form a supported lipid bilayer as shown here. And as you can see, we have transmembrane protein overexpressed that are fluorescent, and we can actually fluorescently observe the lipid bilayer formation. Having said that, typical methods for assessing supported lipid bilayers are optical microscopy based through the use of fluorescent tags. There are mechanical methods as well, AFM or QCM, and electrical methods, which I will focus on in my talk because they're very dynamic and can give us some quantitative results that others cannot. Continuing from the method of making those bilayers, the Daniel team has actually tested with different methods the actual key properties of the cell membranes to reassure that they are performing in their natural environment. For example, the bilayer fluidity, okay, they are fluid, they can move. This technique is called fluorescent recovery after photobleaching. I will not go into details. The proteins are expressed through antibody binding tests. They are functional through functionality tests, and they are oriented on the right way. So it's really a parachute mechanism where they fuse and form a bilayer. So they are not upside up. And of course, here you can see that we can develop membranes of different types of cells. For example, mammalian membranes, virus-like membranes, bacterial membranes, cancer membranes, plant membranes, any cell. We can induce the blebing and develop the membrane of interest. So what we're trying to do, and this is something we developed back when I was a postdoc in Rochenone's group in Cambridge, we are trying to address the key challenge I mentioned before about the importance of studying those membranes in their natural environment, having them mobile and fluid, and expressing the proteins properly. So typical support materials like glass do not allow for the proteins to move for the membrane proteins to move properly, because those membranes are tightly linked on the chip, and so the proteins cannot really move. We are trying to solve this interface phenomena by introducing conducting polymers. So those are electrically conducting materials that are polymeric in nature, so they can essentially provide an extracellular-like environment to the cell membrane while making a device that will be able to transduce the signals coming from the membrane directly. So while the conducting polymer acts like an ECM-like structure, it can hydrate actually, like a sponge. By having said that, the membrane is in their natural aqueous environment, and the proteins can move. And the membrane has the natural fluidity. Another device related attribute is that the cleft resistance that we call is minimal because of the seamless, if you like, integration of the transducer, the conducting polymer, and the membrane. Finally, another additional advantage is that because of P2PSS is transparent, we can also use simultaneous optical and electrical readouts. And here indeed, we can see that using it's not really playing, or let me remove my laser pointer. So yes, so we can see the bright dot here is the actual proteins that are being able to move on those cushion-like hydrated environment using P.PSS, conducting polymer to interface the cell membrane, which was pretty nice to see using optical microscopy methods. And so, of course, we are interested in developing chips, hence the name membrane on a chip, microfabricated devices. And the Saleo team from Stanford did a great job on developing a new type of chip based on P.PSS electrode arrays and transistor arrays, which actually solved some of the problems of other previously microfabricated devices in literature, and allowed for seamless bilayer formation on top of those chips. We could use both microfluidics or wells, and we could accommodate those cell membranes on biologically simulated environments. And the devices, you see, show great stability and uniformity. Okay, so what can we measure from the cell membranes? There are multiple things we can electrically measure. Essentially, we're measuring the ions that can pass through the membrane. We can measure ion channel function, or we can measure, of course, whether there is some, let's say, redistribution of the lipids on the lipid packing that would induce more or less ions to pass. How do we do that? Actually, we have the membrane on top of our device. And if we model it with an equivalent circuit modeling, we can model the electrolyte as a resistor, or conducting polymer through a capacitor, and then the membrane itself, like a cell, it can be simply modeled as a resistor and a capacitor in parallel. Of course, there are other models that can be more reliable, but this is the simplest, which explains, can give us many information already. And here you can see an impedance plot of the device prior to the membrane formation, and when the membrane is formed, you can see this characteristic shape, which is reminiscent of the fact that something very resistive is on top of the device, impeding ion channel motion. And you can see this characteristic semicircle formation on the Nyquist plot. And you can imagine that if something happens to the membrane and it allows more ions to pass, then we would see a change in that direction and that direction simultaneously. And if something happens and it makes the membrane more resistive, more, let's say, it allows less ions to pass, if you like, right? For example, if virus fuses on top or ion channels close, we would expect to see a direction on that side. And through equivalent circuit modeling, we can extract capacitance and the resistance of the membrane, so we can have a quantifiable signal. And with our colleagues in Stanford, we have modeled the response of the device by changing the resistance and the capacitance of the membrane. And so now we know more or less what to expect. And once we can see the response of our device, the biological response of our device, and then through this modeling, we can more or less understand what happened to the membrane, that the resistance, the capacitance changed, and in what direction. And then we can extract some biological information on that. A really intriguing aspect of this technology is its sensitivity. So we can elucidate for the first time. For the first time in the sense that it's not possible with optical methods and, for example, FRAP. Differences between natural membranes and only lipid membranes or artificial membranes, if you like. So you can see that the change in the shape of the impedance plot when we have a natural membrane is completely different than that of the tightly packed artificial membrane. And this is because the lipids are more tightly packed, whereas in the natural membrane, we have natural artifacts, which are the proteins, the ion channels, and so more ions can pass, right? And so the resistance is way lower. We can also see in real time a concentration-dependent effect of a common membrane disruptor as a drug, ethanol. We used it as a model drug, and we tried different concentrations of ethanol, and we could see changes in the membrane resistance over those different concentrations, from pore formation to total membrane disruption after 30% of ethanol, as also reported in literature. And we also tried to look into drugs. For example, we performed ion channel sensing using ATP gated P2X2 channels. So we got the baby hamster kidney cells. We overexpressed P2X2 receptor, which was fluorescently tagged. We could see it optically, and then form the membrane. And then we get the response of the device after we form the membrane, the characteristic semicircle formation. And after addition of ATP, we see a decrease in the resistance, as you can see here, which was did not happen in the same natural membrane that from the same cells that didn't have the overexpressed receptor. A similar work with human embryonic kidney cells and TREC1 potassium-gated ion channel. We tried here to look into the effect of ion channel closing and opening drugs, and we could see again the resistance increasing with when we added a drug that would close the TREC1 ion channels, while the resistance decreased substantially with ion channel openers. We had, of course, first to block the general potassium ion channels and then add the drug. But the results were pretty significant. The device was very sensitive to such changes and wouldn't sense, of course, similar changes with the same natural membrane lacking the receptor. We also used it for pathogen sensing. And so in that case, what we did is we made membranes, natural membranes, on top of our devices, and added that, and those membranes had the receptors that could interact with proteins on viruses, right? So here is sialic acid receptor and influenza virus, and painting the right actual experimental conditions, we could induce a viral fusion, right? And so we could see after viral fusion an increase in the membrane impedance as expected, and thus meaning that the membrane became way more resistive compared to the membrane before, which was not visible with the membranes that lacked the receptor that would interact with the virus. So when we had a natural membrane that lacked the receptor that interacts with the virus, there was no virus binding and fusion. And so I hope I convinced you by now that we have developed a technology where we can solve the problem of interfacing cell membranes with active transducers by using conducting polymers as a soft material that can act as a cushion to accommodate the cell membrane, and then reliably use electronic methods. I showed you impedance, but we can also use transistor-based methods to assess membrane-related events at the first point of contact, the membrane. That can be effect of drugs on certain transmembrane proteins, or host pathogen host membrane interactions. So we can see if a virus can interact with a certain membrane and if it can fuse. So again, some mechanistic understanding on infection. And we have shown you how we can do that using combined optical and electrical methods. And now in my lab, we're trying to expand on this suite of tools to study the membrane by adding AFM as well. Given the fact that it is very sensitive, and we're also really interested to understand what happens also on the mechanical properties of the membranes, as we know that when proteins interact with drugs or with pathogens, or when the cells are stressed, the membranes tend to bend or the mechanical properties change. So what we did is we developed a trap array. So we can trap cells or extracellular vesicles, those membranes that I mentioned. It's a very simple technique to do it using a PTMS mold. And in that work, we did the proof of concept using T cells, so whole cells in that particular study that I'm showing you here. And we tried different drugs that affect the cytoskeleton. So we would expect to decrease the Young's modulus, as shown in literature. So we're able to see differences experimentally. And of course, you can imagine having this array of cells. We use also a certain stage, and we can automate the AFM process. So we can do it. We can do a high throughput, because this is the concept: being able to do high throughput measurements in drug discovery and those in vitro applications. But the problem is, okay, we can automate this part, but we cannot really automate the analysis. Even if we were able to measure quickly, we were not able to actually analyze the results. So we had to go through each raw data, do a model, analyze, and this really took very long. So we decided to use machine learning approaches. And the best way to do that was to use directly the raw data. So the dissipation, the tip deflection data, as you can see. And with this method, we were able actually from the image to extract directly the Young's modulus with excellent actually accuracy. And doing some classification, we were able not only to differentiate between control samples and samples that are treated by a drug, so that where the membrane has changed its mechanical properties, but also to differentiate between different drugs. And this actually minimized errors by a lot. And so now the next step is to do the same methods on the same blebs that we use to study the lipid bilayers and expand our suite of technologies in this area. And by this, I think my time is exactly. I would like to acknowledge first and foremost Roisin Owens and Cambridge University, because a big part of this work was done during my postdoc in Cambridge University. Some newer work in Khalifa University, where I'm now an assistant professor, and my laboratory of biosystems and bioelectronics on a chip, our lab members, the funding agencies, and thank you very much for this invitation to Jonathan and Alex, and to all of you for your attention.
Okay, thank you so much, Anna Maria. Just a reminder, we'll save questions for each of the talks until we finish all three of our youth talks, and then we'll have a panel discussion. So our next speaker is Helen Tran from University of Toronto. Helen, why don't you share your screen, and then I'll introduce you. Okay. So Helen is an assistant professor at the Department of Chemistry, cross-appointed with chemical engineering and applied chemistry at the University of Toronto. She got her PhD at Columbia University and did a postdoc at Stanford. And amongst her numerous awards, she was an AAAS If Then Ambassador, a ACS Polymers Gold Rising Star in polymers, and was a CNEN Talented 12 last year. Helen, looking forward to your talk.
All right. Thank you, Jonathan, for the introduction. I'm very excited to be here. My talk will sort of be sandwiched in between 2 more engineering talks. It'll be quite chemistry focused today. Okay, so my name is Helen. I'm actually, a pretty new group. We started in 2021. These are just pictures of my graduate students. We started in the middle of COVID, so there was 3 or 4 of us, and now it's grown to a group of around 11 graduate students. And so in my group, we are broadly interested in designing new materials that are soft and compatible. Maybe they have sequence control, and can we do this through a lens of sustainability and automation? And today, I'll be sharing some of my work on the sustainability side, specifically on degradable conjugated polymers, and hopefully I can touch base on the other elements in future talks. So you'll hear a beautiful talk by Sihong in the next 30 minutes, but I really do believe that future electronics become even more seamless and functional in the future. We're going to have things that are going to be fun, let's say just some jewelry and light on our skin, but some very beautiful examples that can really improve how we interact with electronics. So there's some nice work on meditation or devices to understand our organs better. And there's a beautiful example from John Rogers' group in using these sort of wearable sensors for ICU babies. There's interest in academia, but also translational applications of these types of materials. And in particular, what I'm really interested in is thinking about how to make biodegradable devices, because I think this will open up a new opportunity. We can think about using electricity as medicine, or having a degradable wireless pacemaker. And ultimately, in these sort of examples, the device will degrade after its time of use, so that you don't need to go and do a secondary surgery to remove the device. As we start developing different types of sensors, let's say for food packaging or tracking, a lot of these materials will go into our environment. So if we can design them so that they are degradable from the onset, we won't have this issue. So to me, I think it's really important to develop these important new chemistries early on so that we don't fall behind the device work. And then lastly, here's another example where you can use a temporary sensor to monitor blood flow. And then after its time of use, it just gets degraded in the body. Ultimately, I think there's some very beautiful examples out there already, but there are limited examples of what type of degradable semiconductors and conductors that are out there. And I think if we can develop new materials with degradability and maybe some other additional functionality, we can even expand to the different types of devices that we can make and make the processing a little bit simpler, as some of these metals could be difficult to work with. So that's what I do in my group. I'm going to actually show all my work when I was a postdoc in one slide, which is always blows my mind. But I worked on imine-based semiconductors, and they're a really nice system because in semiconducting polymers, imine bonds actually maintain a conjugation along the backbone, but they're also labile. So what I mean by that is often for semiconducting polymers, what you want is alternating single and double carbon-carbon bonds. There's other factors, especially with processing and packing and understanding the morphology of these samples, but from a chemistry perspective, that's often what you need to get conjugation. However, these carbon-carbon bonds are quite difficult to break. So if we replace some of the carbon-carbon double bonds with a carbon-nitrogen bond, double bond, what we get is these bonds that maintain conjugation, but they are, with acid and water, they can break up into a dialdehyde and diamine here. And what I was also interested in was trying to make a lot of these types of polymers, but in a modular fashion. That's because a lot of these compounds are readily prepared by, reported literature, and it takes a bit of time to make the dialdehyde. And also for batch-to-batch variability, imine condensation is great because you don't have to use tin, but I found that there were some inconsistencies. So we developed a Stille polymerization, and this was very, very consistent and reliable. Some notable limitations of the imine condensation, like I mentioned, if we have the dialdehyde already, this polymerization happens very quickly. But if you don't, you have to devise new synthetic routes to produce this. And if you are wanting to explore a variety of semiconductors, that could really slow down the pace of research, because if you wanted to use a dibromo compound, a lot of those are even commercially available. And with the the Stille polymerizations, there's quite a bit of tin, which is quite toxic to work with in the preparation of the monomers, as well as the waste, right? So lastly, something that I always wanted to figure out in my postdoc, but was never able to figure out, is how to actually recycle these polymers. We ultimately were unable to figure out what the compound degraded to. So this DPP compound, as soon as you degrade it, this is one of the first projected monomers of byproducts. However, this DPP ring can also ring open. So therefore, we weren't able to recollect or even ultimately understand what our degradation byproducts is. And the same was true for these other thiophene imine compounds, because upon degradation, you get an amine thiophene, which is not stable. So in my group at University of Toronto, we started tackling these sort of different issues and different angles, and are really learning a lot on how to advance semiconducting polymers and use them for degradable applications. So here's my team. There's 4 graduate students in my group who are working on degradable conjugated polymers. I talked about imine condensation, as well as Stille cross-coupling polymerizations. And some work that we recently submitted is using direct arylation polymerization to prepare imine-based semiconductors. And this method is much more atom efficient, as well as it doesn't require the use of tin. So this is work that Nathan really led. And in looking at what monomers we want to work with, we started to use IDT instead of DPP or NDI. And that's because IDT has really high charge carrier mobilities that's comparable to amorphous silicon. And also interesting, I think, like Anna Maria also mentioned this a little bit, some of the work by Saleo. And there's some other work from his group where they report the near amorphous morphology of these samples. So right now we're developing the degradable semiconducting polymers, but ultimately maybe we want to introduce other functionality like such stretchability and elasticity. So, Nathan went ahead and prepared these TIT compounds quite readily. You can make this on gram scale. And then you can also do a simple transformation to make a dibromo compound. And, there's actually quite a bit of research on using DARP for other types of conditions. And there's a lot of variations on DARP as well. So you can really change what type of catalyst or ligand, solvent, or even acid and base that you include. And what we followed was some of Ozawa's work in using these optimized systems here, as I shown here. And what's really nice about these conditions is that what we're trying to avoid is cross-coupling on DARP. So when we first tried these conditions, which worked well for IDT-BT, we switched out the BT for our dibromo TIT compound. We actually found that it didn't work very well. So it's not so easy and as I expected. So the molecular weight was quite low. We get somewhere around 3 kilograms per mole. You see a slight color change upon polymerization, and when you try to collect it, you get some sort of material that's really difficult to precipitate out. So what Nathan did was he started looking at what's happening in NMR, and you can even see that the signals are quite messy. So if you were to look at some EPP, the NMR might not be very informative, but for IDT, you can actually see individual peaks that can give you information about whether cross-coupling is happening. So there's a lot of optimization that's needed here. So Nathan went and dug deeper into literature and looked at some of other Ozawa's work and found that sometimes the addition of timida can actually, as a co-ligand, can actually increase the molecular weight and yield. And part of that is reducing the undesired cross-coupling products that are often insoluble. So by adding some timida for these compounds, they were able to increase their yield up to 80% and 20 kilograms per mole. So this is promising work to show, without timida, they get around 3K, and that's actually quite similar to what we got. As soon as we start adding timida, you can really push the molecular weight up to the 20K region. So that's exactly what Nathan did, and it worked quite beautifully as well. You can now see a really nice color change. Without timida, it remains this reddish color, and with timida, it's around this purple color. And we can even collect up to 28% of the material in the hexanes fraction as well. And then if we continue to do a second oxalate in chlorobenzene, we can collect some polymer that's actually 25 kilograms per mole. Okay. And now if we look at the NMR of this polymer, you can see that the baseline is much cleaner, and you don't see all those additional peaks that we saw for the no timida section. And this really lines up nicely with our predicted structure as well. Looking at the imine peak itself, you can see that if you start with IDT, we don't have an imine peak here. Our dibromo TIT has a peak around 8.2 parts per million. And as soon as it becomes a polymer, you can see this really nice downfield shift as expected. Now, in this polymer system, another handle that we looked into was actually concentration. So, if you have a more dilute sample and you run it for a longer amount of time, we were unable to get high molecular weights. And so going from 0.05 molar up to 0.25 molar, we actually saw much more consistent results in yields as well. And what's really interesting about this system compared to all the previous systems that I've worked in is actually doping. And so for this IDT-TIT compound, we found that upon adding some acid, we can go from the benzoid to quinoidal structure here. So in the previous DPP-based systems, we wouldn't see this doping shift. You immediately see a degradation and ring opening of the DPP. But here, since there's no ring opening within the IDT compound, we see this really nice doping that can be observed by UV vis. So we added TFA at different volume percentages. So starting from no TFA, as soon as we add TFA, we see this peak slowly disappear and the emergence of a peak around 800, which we are referring to the dope peak. And if we actually do this experiment, we have to really carefully add the amount of acid. If you just take the same sample and add more acid over time, that's another factor that will come into play as well. And so here, if we take the same concentration of acid on the sample and just monitor it over time, we can actually see the decrease of the UV vis. And this decrease is expected because as the polymer is sitting there over time, it will start degrading the imine bond. So for low concentrations, what you see is an immediate slow degradation of the polymer. However, in the high concentration of TFA, first you see the drastic shift to a dope state and then slow decrease over days right here. You can see really nice colors associated with this color change as well. We also observed this in thin films. So here is some solution UV vis spectra before and after TFA addition. And for the thin films, what we did is we placed a glass chip inside a cuvette and added a drop of TFA, and we can actually see pretty similar shifts as well. So going back to the degradation aspect, one thing I really wanted to do was recollect the monomer. So one thing we noticed is just placing the sample for a little bit over 2 weeks, regardless of the concentration, we found that all the peaks sort of coalesced towards around 475 nanometers for the degraded product. So we were really hoping that since there's no ring opening, we just break this imine bond, that we get this byproduct that has IDT and then thiophene flanked, where one side is an aldehyde and the other side is an amine. However, we were unable to identify this compound, and it really goes back to the stability of this thiophene amine. So Shine went and made this compound here that's serving as our model to sort of see where the shifts are. And it gives us a boundary limit. So this is your IDT TIT compound, IDT flank with thiophene, and this is our starting material, and our degradation product is somewhere in the middle. So we have something that is a little bit more electron withdrawing compared to this compound. So even though we were unable to get the monomer, that really made us think about what amine needs to be made so that it is much more stable. So this is some work that Azalea did in collaboration with Minoru Ashizawa, as well as his student Naoya Nozaki, sorry. And so previously, if we were looking at these DPP-based systems, what we have is ability to polymerize the polymer, but unable to recollect the monomers. So we devised a new TII compound where now we have the dialdehyde also on this compound and a diamine. However, we have a different system where we hope that ring opening wouldn't happen. We find that similarly, just using imine condensation, these polymers are readily prepared. You can see a really nice color change going from blue to green. And as soon as you add TFA, you revert the color back because you're moving back to the starting materials. And if you add TFA, again, we actually see a really nice shift in the doping immediately in the maximum of the UV vis. As you place that sample over time, the UV vis maximum also decreases. And what's happening here is we also get no ring opening, and that allows us to dope our system and create this stable intermediate so that we can see this new dope peak. And because of this imine condensation, our diamine is now on the phenyl, and our dialdehyde does not have a ring opening labile compound. We can actually recollect the monomer. So here you have in purple, the top line is our pure starting dialdehyde. If we put it in TFA for 24 hours, the UV vis stays effectively the same. And even after 6 months, the UV vis is pretty much similar. We can actually recollect this in quite high yields, around 90 to 95%. And if you compare the NMR spectra for before and after of the dialdehyde, they're identical. And that's also true for the mass spec as well. So we're pretty confident that we're recollecting our monomers. And if you actually take these polymers and cast the thin films, we can look at the morphology of these samples, which I really want to do more in my group, which I'll talk about at the end. We can actually see slight differences as we vary what our diamine is here. So unlike our previous work, some of our collaborators were able to make some devices for these for these samples, and we see sort of characteristic behaviors. I think there's a huge opportunity to improve the mobility, but what we notice is going from the pristine to the recycled system, the performance can be maintained or actually improved. And so the last part of my talk, for the remaining 10 minutes that I want to talk about, is also, beyond sort of getting a better understanding of the molecular way and how to control the degradation byproducts and polymerizing with new techniques that are more atom efficient and tin-free. We want to think about new types of monomers that can be explored. And so we're looking at bio-based compounds, and I completely recognize not all bio-based compounds are non-toxic, but we want to look at things that we actually find in food and things around us. And there's a lot of compounds in nature with known degradation pathways, such as chlorophylls, carotenoids, betadines, and curcumins in the food that we eat. And in my group, we're really excited about carotenoids because, the backbone of this compound looks very similar to polyacetylene, which won the Nobel Prize in 2000. And, if you look deep into carotenoid research as well, you can actually find some very beautiful work. If you look at a carotenoid diacid, so called Bixin, so this one derivative, if you just take that small molecule and make a field effect transistor with it, you actually can see some nice characteristics. The mobility is quite low, it's 10 to the minus 6. We really want this up to 10 to the minus 1 or 0.1, even 1, right? But this is a promising start for this compound. And if you take keratone analogs and do single molecule conductivity studies with AFM, you get very high mobilities. And that's sort of expected because these carotenoids are effectively polyacetylene monomers, right? And lastly, we really like carotenoids because they have really interesting degradation pathways that are known. We can cleave a lot of our imine bonds with acid, but there's some enzymes that selectively can identify this particular carotenoid bond so that it can degrade into retinol and other byproducts. However, right now for carotenoids, even though there's some electronic behaviors and the monomers could be readily available, there's a really poor understanding of the morphology and the solubility of these compounds, and there's no polymer version studied. So in my group, what we're trying to do was figure out how to use carotenoids as a repeat unit in our polymer. If we take why do we want to make it into a polymer? If you go from a small molecule monomer into a polymer, what we can do is effectively reduce the band gap. And if we add solubilizing side chain groups, we can take something that's often not soluble and make it soluble. And we can really learn a lot from the organic solar cell community on how to do this types of side chain engineering. So we can add just simple hexyl groups, but even branching points and where the branching points is introduced, all of that could really influence the morphology of the thin films as well as the effective field effect transistor mobility at the end. So this is work that Azalea did. She went in and looked at imine condensation of a p-phenyl-diamine derivative and copolymerized it with a C10 aldehyde that is found is a carotenoid derivative. She made a model compound. She made an insoluble compound and also started adding methyl and hexyl groups to see if we can make these polymers much more soluble. And the answer is yes. Azalea, I think this is the first experiment that Azalea did. One of the first experiments she did in the group where she took her carotenoid and diamine and polymerized the compound. And she made this black solution that came out as a black solid that was not soluble in anything that she could try. And that was exactly what I wanted to see because that was our hypothesis. And as soon as she started adding methyl groups and hexyl groups into the side chains of the diamine, you can actually see that black powder slowly become much more soluble as you go from the methyl to the hexyl group. And we see the solubility change differences in chloroform and THF. And what we wanted to do was quantitate how much better is the hexyl group compared to the methyl group. So Azalea did a series of UV-vis experiments and measured them against a supersaturated solution. So first, she created a series of controls with different types of concentrations, created the supersaturated solution, and to figure out what it was, she just diluted it until it fell within the range of the standards. And she can back calculate it that the saturated concentration of the methyl group is actually 34 milligrams per milliliter. However, for the hexyl group, it actually was 67 milligrams per milliliter. So this quantitatively told us that moving from methyl to hexyl was a big improvement in solubility. Going from the C10 dialdehyde to the model compound to the hexyl group, we were able to extend the conjugation along the backbone and saw the corresponding UV vis shift as well, seen here. And as Alea started to do some degradation studies, we also see the opposite trend, right? So first she started with this black trace here. As she added more acid, higher concentrations, it shifts back to the left side here, which this left trace resembled the C10 dialdehyde starting material of our monomer. And lastly, Alea was really interested in even exploring how light can influence the degradation of these materials. We know that without light, the acid has an impact on the degradation of the polymer. So you can see the initial red color turns yellow in the end. If you don't have acid, you see a slight decrease. That's hard to see colorimetrically, but you can see in the UV vis. And one really distinct example is actually seeing with acid and with light, you can see that the degradation is enhanced with light compared to the dark. And really to end, what I want to say is, Alea is doing a lot of work on these compounds now, especially on the doping side, which I didn't include in today, and really understanding how to design these carotenoid-based polymers again, so that they can even be better. So we were pretty happy to get this red powder that was our carotenoid polymers that I talked about in the previous slides, but some initial work that Alea has already shown by just tweaking the molecular design of the polymers, we can get this really nice black polymer now that you can visibly feel is quite spongy and much softer, which is an indication to me that the molecular weight is very high. So with that, I want to conclude that that's sort of the efforts that we're doing in my group in terms of sustainability. And eventually what we're looking into is moving into making these polymers a lot longer, softer, and stretchable, and automating a lot of these types of syntheses. With that, I'd like to thank my group. I feel very fortunate to have such a nice group to work with. It's been really hard, I think, for many of the group members working in a place where, we don't have a lab, but we have a lab now. And, working together has been really beneficial for them to learn a lot of synthesis and eventually some device work as we get some of our equipment. I also want to thank our collaborators in this area, specifically Minoru and his student. We're also doing some computational work with Mario Leclerc and his student Tristan. And lastly, I'd like to thank all our funding, particularly NSERC, CFI, U of T, the Acceleration Consortium. And thank you all for your attention. I'm going to stop sharing.
Thank you so much, Helen. I have a few questions for you right now, but I'll resist the urge and I'll ask them. I'll ask them later when we have our panel. Fantastic. All right. So that's two talks, and our third this morning is from Sihong Wang from University of Chicago. Let's share your screen, and then I'll introduce you. Okay.
Yeah. So is it showing right, Jonathan?
Yeah, it's looking good. Let me just briefly introduce you. So Sihong Wang is an assistant professor of molecular engineering in the Pritzker School of Molecular Engineering at the University of Chicago. He did his training at Georgia Tech, his PhD. His postdoc was at Stanford, and now he's at joint between UChicago and Argonne. He has a number of awards. Let me pick a few here. He was also a Technology Review 35 under 35 from MIT Technology Review in 2020. He's a highly cited researcher. He was an ICANX Young Scientist Award winner in 2001, and lots of other awards. But in order for you to have time to share your great results, I'll let you take it away.
Yeah, thank you so much, Jonathan, for the very kind introduction. And thank you for mentioning the ICANX Young Scientist Award. That's one of the favorites, my favorite awards that I have received so far. And similarly, I'm very appreciative of this, really fantastic ICANX platform. So I just counted. This probably is the fourth time I give a talk in this platform. So today, hopefully, I will, share with you about some like our recently published works from the time like about 6 months ago that I gave my last ICANX talks. So, all right, so let's still, start from the broad like motivation of our research. So, in general, what we work on is highly aligned with today's topic, bioelectronics. So bioelectronics basically is capturing the use of electronics that need to directly interface with our human body for a number of emerging application areas that has just come up in the recent decades. For example, like precision medicine and the Internet of Things, and especially if we take our human body as the most precious things to each of us, and also human robots collaborations, human machine interactions. So for all these needs, the current form of electronics that has been developed mainly based on rigid and brittle inorganic materials has significant mismatch in its mechanical property, in its general geometry, and also the chemical compatibility with our soft, constantly moving, and curvilinear human body. So therefore, for these new electronic application areas to really achieve the most effective communication and integration between the electronics and the human body, what we are poised to do is to develop new type of electronic devices, or even starting from materials that can really mimic different kind of properties, physical, chemical properties of the biological system of the human body, so as to achieve the intimate integration and even finally the seamless merging. So as the previous speaker already mentioned, right, among all the different kind of materials, polymers actually can be a very special and especially useful one for this kind of electronics developments. So then the first question we will ask is why polymers, right? What's so unique about polymers? So if we, first take a look at the chemical composition of our own human body, we will realize, right, besides most of part, of course, is made up by water, the rest are like predominantly like composed of polymers and other organics. So then basically it means polymer chemically is already inherently compatible, similar with the human body. And then the polymer also, like compared to inorganic materials, they are naturally flexible and can also be made stretchable. So that's another advantage. And the polymers are also like has a move-free volumes. So then they can be made into membranes for allow the transport of different species, for example, biomolecules, or even be made with ion conducting properties. So that's another advantage that can enable more effective communication with biological information. And also for polymers, like Helen just showed, it has a lot of very broad room for chemically tune its molecular structure by changing the backbone or the side chain. And you can do it in a fairly decoupled way. So basically it means that you can play with the structure of a polymer like a Lego. So then it also gives the space for us to like integrate different kind of properties together on the same material. So then overall, the research that my group has been pursuing is to develop this so-called new type of a biomimetic polymer electronics that can be integrated with the human body, either on the skin as wearable devices or implantable, or with the organ or tissue as implantable devices. So our goal is to develop this kind of electronics that has functions across sensing of different biochemical signals, biological signals, and then process these signals using like machine learning and AI, and then also use the process information to further like modulate or feedback to the human use. So the overarching goal that we work on to start from the very basic material level to develop the new type of biomimetic functional polymers, and then use these to make devices, is to realize really the continuous, efficient, and also long-term stable acquisition and the processing of health data. So today, in this talk, I will be mainly talking about the work we have done in the sensing parts. And if there is a time, I will just very briefly mention our work in the modulation, particularly the development of OLED, and then not really talking about other things. But if you are interested, here are some recent representative papers from our group. All right, so then let's start from biosensing. So when we are working on biosensing, the question that we keep asking to ourselves is first, what is biosensing? What is a bioelectronic sensing device? What does it do? And the second, what constitutes a good or say high performance sensing device? That basically is a direction that we need to put our effort on. So regarding the first question, sensing function is actually relatively obvious, right? It is the kind of device that take bio signals from the tissue and then converts to a different type of electrical readouts so that you can record, you can analyze. And then for this basic property, the high sensing performance is largely coming from the property of the electrical readouts you can get. So overall, high performance sensing can generate high signal to noise ratio for the electrical readouts. So therefore, the overall developments of the bioelectronic sensing, biointerface sensing, is about how we can engineer the device or even the way it integrates with the biological system to really enhance the transduction of the signal to the final electrical readouts. So then to think about this, we need to break it down a little bit, because this is not just a one-step process. It may involve different things. So what are they? So we think there are overall two steps that are very important. First, the biological signal, when it's generated from tissue, it needs to be first received by the sensors, need to get onto the sensors for it to be sensed. If it's not getting onto the sensors, no sensing can happen. So this is first step. And then after the sensor receives the signal, now it's really the device taking the function to convert it to electrical readouts. So that is the sensing of the device. So therefore, for the high performance sensing, we need to optimize or enhance both of these two steps. So then the next level of question is what determines the efficiency of each of these steps. So for the second step, since it's mainly happening on the sensor device, then it's very obvious, right? We need the sensing device with high sensitivity. So that's one aspect. But what about the first step? So the first step actually is determined by the property of the interface between the device and the tissue. So the overall goal is the overall requirements is this interface need to be as conformable as possible so as to minimize the distance and the impedance resulting signal decay, right? Signal decay can happen at this interface, but we want to the maximum possibility, prevent it. So then this drives the overall direction of our research. To get started, right, from this general question, we first, picked a high performance sensing device platform to achieve the overall high performance sensing function. So what we have worked on is so-called organic electrochemical transistors. So it is based on a very unique type of conducting polymer that can be conducting not only to electrons, but also to ions, so-called mixed conducting polymers, so that the externally applied electrical potential can drive the electrochemical doping or de-doping reaction in the semiconducting polymer, which involve both the ion and the electron activity. So the benefit of this kind of a volumetric doping-based transistor compared to field effect transistor is it can provide much higher capacitance for the modulation of the charge transports. And then the capacitance basically give rise to much higher transconductance. Transconductance is the capability of the externally applied gate voltage to modulate the change of the drain currents. So then you can imagine when the gate current modulation comes from a certain type of target signal to be detected, then you can very sensitively monitor the change of the signal from the readout of the currents. So therefore, this type of OECT sensing platform gives a number of very important advantages. For example, it can have very high sensitivity for the reason that I just talked about. And also, the design of the device allows use for both bioelectrical and biochemical sensing. So during the past, the past 10 years, there has been many research groups. For example, our moderator, Jonathan Rivney, has done really seminal and pioneering research that's set the foundation of this kind of a device for the use of a different type of advanced sensing function. So our research is basically largely built upon their work. So the next question that we also want to gain better understanding is for the sensing to happen. Things we talk about, right? The most important part of the sensor is where it interfaces with the biological tissue. So then what we also need to get better understanding what constitutes as this interface of the sensor with the tissue. So from the previous work of using the OECT sensor for both electrophysiological sensing and the biochemical sensing, it has been shown that the semiconducting channel is probably the most effective way to collect the signal from the biological system and perform the sensing function. So therefore, it means that the most important material, the key material in the sensor is the semiconducting polymer. So driven by this overall understanding, the first thing that our research gets started is to think about when we utilize semiconducting polymer for performing the sensing, especially our interest is largely in the biochemical sensing, then what are the material design requirements for the semiconducting polymer? So the thing we start to realize is for biochemical sensing, in order to realize the selective or the specific sensing function, usually the sensing material need to couple with a certain type of biorecognition units so it can have the selective binding to the analyte targets. So then the semiconducting polymer need to be designed and synthesized in a way that's it has versatile functionalization of, for example, DNA molecules or antibody molecules on its chemical structure. But this hasn't been really a mature avenue to do this in the past. So then what is the challenge? The main challenge is, as you have seen from Helen's talk, there have been, for the synthesis of a conjugated polymer, it's overall a relatively complicated process. So then you can imagine if you need to, synthesize a different structure from the very beginning and then polymerize at the ends, then the condition of the polymerization probably is already not compatible for the biomolecule units that you have incorporated onto its chemical structure. So then to solve this issue, my student Nan Li has came up with the idea of using a click chemistry as a step that happens after you polymerize and get the polymer. So the way he designed this process is to just use one azide unit to incorporate during the monomer synthesis stage, and then polymerize the polymer so that we can get a polymer precursor. And then this precursor will allow just one step click reaction for the functionalization of very many different types of functional units onto the side chain. So then this makes the synthesis on one hand can happen at a very mild condition to broaden its compatibility for different structures. And on the other hand, can also, make the synthesis much simpler. So then using this process, Nan has been able to covalently functionalize an enzyme molecule that is for glucose covalently onto the side chain of this semiconducting polymer that can work as OECT channel. And then when it's used really in the OECT device, the presence of the glucose now can be oxidized by the enzyme, and then induce the de-doping reaction of the channel, of the semiconducting channel, and from which you will see the gradual decrease of the currents. So that's basically showing the stepwise increase of the glucose concentration. So this basically has opened the door of the engineering of the semiconducting polymer to make it more useful for the biological interface, biological sensing interface. And then now we really start to answer the question that we posed previously. That is, for achieving the conformable device to a tissue interface, what are the requirements? So tissue are basically like shown by these two representative examples, brain and hearts. They are highly curvilinear and soft, and also constant deforming, generating a lot of strain. So then, for the deforming aspect, the requirement on the electronic material and device is relatively obvious, right? We need the device to be stretchable so that it can accommodate such deformation. So in the past couple of years, there have been a lot of work going on for achieving the so-called stretchable electronics. This is something people have done tremendous progress. But stretchable is not just the only thing. There are also some other properties also very important. So what are they? The second property actually is the other very important mechanical aspect. That is the softness, or how easy a material or device can deform. So why this is important? From previous research, it has been shown both theoretically and experimentally that the capability or the ease of device to conform to a very curvilinear surface is largely influenced by this parameter, that is stiffness or the Young's modulus of the structure. And then the lower Young's modulus will give you both lower bending energy and also the contact pressure between the device and the surface. Both means it will give you easier conforming. So therefore, another property that's very important is about this parameter, that is to have ultra soft property of the electronic device. Okay, this is the second thing. Anything else? So, let me remind everyone, our goal is to achieve conformable and stable interfacing between the device and the tissue surface. So then, from everyone's life experience, right, for two things to adhere very, very stably with each other, then there usually need to have some adhesion interactions in between, right? Otherwise, if no adhesion, things can easily separate and drift away from each other. So therefore, the device also need to be bioadhesive. All right. So these are the three properties that we think are all very important. Let me start from the stretchable property first. So regarding the stretchable requirements on the semiconducting material, the essential challenge actually that comes from the very different type of material structure, crystalline structure of a semiconducting polymer, which usually need to be in a relatively ordered and a closely packed structure to enable efficient charge transport, compared to the elastomer, like a rubber type of material that is usually built upon very disordered and loosely packed structure that can allow the easy deformation of polymer chains and the stretching. So then the key challenge is how can we consolidate these two very different type of material design requirements on the same piece of material. So in the past, this was several years ago from my postdoc work at Stanford with Zhen and Bao. So we have focused on the design of a semiconducting polymer that has stretchable property for few effect transistors and created the design that uses a physical engineering method. But now we are when working at OECT, we are facing a new problem. That is the semiconducting polymer not only need to have electron transport property, it also needs to enable the efficient ion conducting property and also further have the redox active reaction. So then overall the design of the semiconducting polymer need to have more considerations on its functional side. And then the mechanical stretchability requirements need to couple together. So our idea about how to design this stretchable property is actually starting from how the different polymer chains interact with each other. So we think if we can really weaken the interaction between the polymer chains, then during the deformation, the different polymer chain probably can more easily slide against each other and reorient so that the strain can be more effectively dissipated. So Then this leads us to the identification of this polymer structure that has an asymmetric backbone. So that's the pipette stacking between different polymer chains are not as strong as a symmetric structure. So this polymer, after we synthesize and test it, it really enables us to create the fully stretchable OECT biosensor that can perform the electrophysiological sensing, for example, ECG sensing in this case, on the human body. So we can see when it's on the wrist, it can deform together, but without influencing its property of reading out the ECG signal. So this is a first step about the stretchable property. But as I mentioned, right, stretchable is not the only thing. The other very important requirements is the stiffness. And if we compare the Young's modulus of the brain and heart to the stretchable material and devices, especially semiconductors, we can see there is a still large mechanical mismatch in between. But if we take a broader look at the synthetic materials, basically the only type of material that can give the tissue-like softness are very limited. It's mostly just hydrogels and some other ultra soft type of silicon elastomers. So then it means we need some new electronic design ideas that can allow us to use such a very soft but non-electronic materials to realize the electronic devices with a matched stiffness with the tissue. So then this, probably can enable us to really achieve the better conformability on the skin and tissue, and also very importantly, for the implantable application, it can reduce the invasive reaction and the foreign body response of the devices. So then what are the possible strategies? Actually, the initial thing we started to think about is this probably is a pretty simple challenge to solve, because the overall mechanical property of a device is largely determined by its substrates, and the substrate doesn't need to have electronic function. So then, can we still use those high modulus but stretchable electronic materials to combine with the soft substrates to build device? And then when we start to do some very simple tests, we found actually there is a very major challenge. That is, when there is a significant mechanical mismatch at the interface, it usually will lead to significant concentration of stress. That will lead to a large decrease of the stretchability of the supported high modulus film. So then, to address this challenge, so that this general design idea of combining high modulus electronic films with a low modulus substrate still can be possible. My student Yang Li has came up with an idea, very simple, but has a universal applicability of inserting a soft interlayer that has both intermediate modulus between the high modulus thin film and the soft substrate, and also can form good adhesion to both sides to buffer the mechanical stress in between. And Yang has found that actually a very commonly used elastomer, so-called SCBS, can do this function very well. So then, using this design, by inserting the SBS into the interlayer as an interlayer between the stretchable semiconductor and ultra-soft substrates, now with the gradual increase of the soft interlayer thickness, we can observe a very effective improvement of the stretchability. The crack gradually goes away, and then when it's tested in the transistor device, we can see also the stability of the mobility also gets significantly improved. And then when using this method to create the sophisticated integrated circuit, now it can allow the circuit to be much more softer. Basically, it's built on hydrogel and has a similar modulus with a hydrogel, but can perform in a highly stretchable fashion. And with this device, now we have started to test its advantage. So for the foreign body response that I have mentioned earlier, we have shown that by comparing with hydrogel, this bilayer design with the added soft interlayer can indeed keep the same level of foreign body response, which is much lower than just using the SEBS, although it's an elastomer, but with higher modulus as a substrate for the device. And then, on the other hand, when we use the device to directly interface with the tissue, for example, on the beating heart, this is red heart. Now the device can maintain much more stable during the heart beating, not really drifting after several minutes. That is as shown by this SCBS-based device, and also causing like minimum influence on the heart's operation function. For example, for this ventricle pressure, for the ultrasound device, the heart can beat at a similar level of pressure as compared to the more rigid device, right? The hearts have to beat harder to like accommodate the additional mechanical force. So this is the second aspect, but this is still not everything. The third thing that's also needed is that to really ensure the good contact, effective and stable contact between the tissue and the device, the adhesion is also an indispensable property. So regarding the adhesion, there has been some conventional strategies for bonding the device to the tissue surface. For example, either by suturing or stitching of wires or staples around the edge of the device, or applying non-electrical adhesives in between. But both of these strategies have their own limitations in the invasiveness and also preventing the real like direct contact. So then our thinking is probably the most desirable way is to create the direct adhesion between the sensing layer, the semiconducting polymer, for example, and the tissue surface. So this probably need to require like new designs of semiconducting polymer to make it really bioadhesive to the tissue surface. So then what are the possible strategies? Our initial thinking is very simple. So we need two functions, right? One is a bioadhesive, the other is semiconducting property. Then how about we take two different kind of polymers and then blend them together to form this kind of a double network structure, so that each polymer still provide its own function. So then two functions can be added up together. So then we started off to try this idea. So we still utilize the semiconducting polymer that we have studied before, and then combine with some bioadhesive polymer that people have already reported in the past. For example, in these two representative works, so they are designed in a way that they have bioadhesive groups to form covalent and non-covalent interaction with the tissue surface. But we found that after blending them together, actually, the initially high adhesive from this kind of bioadhesive mimicking polymer now decrease a lot after blending with semiconducting polymer. So then we start to think, what is the reason? What is causing this substantial decrease? And from looking at this polymer structure closer, actually, my student Nandi noticed a potential issue that comes from this very long side chain of the semiconducting polymer, which may like screen and shield the effective exposure of the adhesive group at the surface of the film. So then to address this issue, he designed a structure that's significantly extends the length of the side chain so that it can enable the effective exposure of the adhesive group. So we call it a brush adhesive polymer. And then also this design has relatively hydrophilic property. So it can also temporarily absorb water or fluid at the tissue surface to like drain the tissue surface so that the direct physical contact between the adhesive group and the tissue surface can be realized. And also is a highly soft and viscoelastic. And now using this new design of a bioadhesive polymer to mix with a semiconducting polymer, now it gives like the continuous double network structure to allow the effective charge transport. So then using this design, now the adhesive property can keep at a similar level with a bioadhesive polymer and significantly improved compared to the semiconducting polymer by itself. And we have also shown that the adhesive property can be well achieved on different kind of wet tissue surfaces. And then as a semiconducting material, we also tested its performance in OECT and show that for both the mobility and the transconductance, it can maintain at a similar level compared to the semiconducting polymer itself. And using this material, we have further made the fully adhesive, fully bioadhesive, and stretchable OECT biosensor that can be very easily attached onto the hard surface to achieve the sensing function by just simple pressing, very briefly, like a gentle pressing. And then it can maintain stable contact on the heart. When even there is like a small pulling force, it won't drift away. And then the stable recording of ECG signal can be kept. And in comparison, right, when we are using a device that doesn't have this kind of a adhesive property, everything else is similar. Initially, it can not form a good adhesion, conformable contact. And when there is a force applied, it will gradually drift away and then eventually detach from the heart. So this really shows the benefits of using the adhesion property to achieve the conformable interface and maintain the stable contact between the device and the tissue. All right, so I'm seeing my time is running out, so probably I will skip the last parts that I initially planned to talk about, and then directly go to the summary parts. So hopefully I have used the past 30 minutes also to show you some new possibility in designing electronics, in incorporating both stretchable and the bioadhesive as well as the ultra soft property all together, with the overall goal of achieving the intimate and the stable biointerface. So, and at the end, I'd like to acknowledge all the very hard and creative work of all the students, postdoc in my group. They are really the true heroes to make all this happening. I'm only presenting on their behalf, and also all my collaborators and the funding support. And thank you all for your attention. Thank you very much.
All right, thank you so much for that great talk. And thank you to all three of the speakers. So now we are open for the panel discussion. Again, please, we'll ask some questions and get the discussion going, but please, if you have any questions, please enter them into the chat. Fantastic. So I'll reintroduce Alexandra Rutz from Washington University in St. Louis. Alex, please feel free to introduce yourself, and we'll get the discussion started. And we'll ask Helen and Anna Maria and Sihong to please turn on their video so we can have a lively discussion.
Thanks, Jonathan. Congratulations to all of the speakers. Really fantastic talks, and thanks so much for showcasing all of your great work. So I'd like to start off with the first question. It's directed to all of you. Thanks so much for going over your work with showcasing the great benefits of conjugated polymers for various biointerfacing applications. I wanted to ask you, where do you see the future of these materials? Are there other applications besides bioelectronics that you see these really being impactful? And additionally, where do you see the remaining needs for these materials to be competitive to metal and silicon-based conductors that are really dominating our current lifestyles?
Do I start?
Whatever order, go for it.
Okay, perfect. So thank you so much again. It was really insightful to also hear to the Helen's and Sihong's talks. So for me, I'm not a chemist. So in terms of materials, chemistry, I could not really say a lot. But as a big picture, what I see is the main challenge, if you like, for those materials to be translated, let's say, because there are a lot of proof of concept works in literature and very impactful works, actually, which really show in animal samples, cell samples, on skin applications, human even samples. They really show the potential, and the fact that they do outperform conventional materials. And they outperform conventional materials not necessarily because their electronic properties are better, it's because of this mixed ionic electronic conductivity and their ability to actually conform to tissue and form a better interface. So they do outperform state of the art. However, the problem I see is that they are not really standardized. the methods we use to make them or the methods we use to test them, they're not standardized yet. So as different groups use different metrics and figures of merit, I mean, Jonathan has done a great work to introduce some figures of merit for the characterization of those materials. But when it comes to their interface with biology, a lot of studies have happened, but there is not really a standardization. So this is where I see that we need to focus more is to develop some figures of merit with regards to their interactions with biology and standardize a bit more the characterization of those materials in simulated environment and get a universal, let's say, a way of doing it so that we can ultimately translate it to clinic.
How about we go in order? Helen, what are your thoughts?
Well, I guess as the chemist, I'll have to comment on the materials question. I mean, I think magnesium and other types of metals are great, right? There's sort of nothing wrong with them. It's more like it's not one material can be used for everything. If you use a different material, then it could have properties that the metal doesn't have. Sihong sort of discussed this a lot in his talk, and Jonathan and others have also worked in this area. If you use something that has both ionic and electronic conduction, you can make new types of devices that are otherwise not possible. And for me, I would just like reiterate what Sihong said, like polymers are really interesting because right now the electronic performance is not as good, but it has all these really interesting mechanical properties that are otherwise difficult to engineer. So it's not, I actually think metals and polymers are quite complementary, and there's probably some opportunities to integrate more of that as well. And I think we're all thinking about that right now, but I don't think anyone really sees it as a metals versus polymers, and they each have their own applications.
Mm-hmm.
Yeah, maybe I can also, add some more perspectives from my, my thinking. So I fully agree with what Helen and Maria just mentioned. So indeed, right, it's very exciting to see, how much the community has gradually contributed from different aspects to lead like polymer as electronic material to what it can do today. And it's probably thinking towards the future, it will have like kind of a very complementary function with the traditional like silicon and metal-based electronic materials. And then for the polymer, overall, I think the main advantage probably will be in the like interfacing with the biological systems, using its like a soft property and a highly modulatable chemical nature. But there are indeed more things that still need to be done. For example, right now there are still not enough understanding about how tissue or the human body, the immune system react to the different properties, chemical structure of a polymers. So overall, if you want to realize long-term like stable functions without too much inflammation reaction, that is also very important knowledge that need to be studied. And also how to maintain the like stable chemical property of the like polymer in the human body environments, especially when it needs to be exposed on the surface to have direct sensing property happening. So human body is very complicated, and in some cases also like very unstable kind of environments. So those are new challenges that still can enable us to do really breakthrough works.
Great. Maybe a follow-up question to that is, especially for Ana Maria and Sihong, you talked about technologies that you're developing, right? You talked about sensing diagnostic platforms, or platforms for fundamental research, or devices for in vivo implementation. What are those challenges for your particular technologies in order to really take them from this proof of concept stage to a product, to something that can go into the clinic, to something that can be deployed for diagnostic purposes?
Okay, I guess we follow the same order. Yes, that's a really nice question. What I face a lot during what I have faced a lot during my different works using, I work mostly with commercially available polymers. So I didn't work a lot with polymers that have been synthesized by groups. So we have a material that is being, that is standard. And we buy it, we get it. The problem is that when we process it on a device, when we make the platform, we don't always get the device to device, batch to batch, user to user. We don't get very small variations. Sometimes we get larger variations. So the devices themselves are not really standardized. Other groups use this type of fabrication, other groups use another type of fabrication, different characterization methods. So from university to university, because we work across different universities, we get sometimes different results. So it has been really, really difficult to be able to standardize our processes in a way that when I do my experiment in Cambridge, I will get the same data in Stanford, in Khalifa, in Cornell. That's an additional problem when you have the biology on top, right? Which is also very unpredictable. So when we talk about standard materials that we can use, because of course, if there are new materials, there needs to be a whole new process to be done to standardize them. I guess what is really important is to standardize the fabrication, the device integration, the biology, the packaging, all those aspects, so that they can reliably operate with smaller variations, and so that they can be used actually for next stage of more, let's say, clinical related trials and studies.
Great. What about you, Sihong? What are the biggest challenges for translation of your technologies?
Yeah, thanks, Jonathan. Yeah, I first want to, strongly echo what Anna Maria just mentioned, right? Like the standardize and also like us minimizing the batch-to-batch variation is also a highly important need that I see from, the things that we have worked on. And then besides that, another challenge that I think also need to be addressed is how to maintain the stable function of a sensor over time, right? Like for example, for using sensor to detect biomolecular signal, usually you want a sensor to detect over a relatively long period of time. And then if there is some overall over time baseline drifting, then it may make the sensitivity or the detection of very small signal basically not so feasible. So that involves a lot of consideration at both the material level and also device level, so that it's not influenced by, for example, the change of the ionic strengths or even mechanical motions around the sensor, but just by the presence of the signal that you want to detect. And then another thing that we have also started to think about, I haven't spent much time to talk about in that aspect, is usually you do not need just one sensor. You need a large number of sensors to perform either multi-modal or the spatially resolved sensing of a certain type of signal. Then that also poses a challenge to the fabrication, especially fabrication using those non-conventional type of electronic, polymer-based materials. So then more work also need to be done in this front.
Lots of challenges. That's good.
Yeah, we like challenges.
Helen, I'd like to ask you a question regarding thinking about the degradability of polymers. So you gave a very nice overview of thinking about the future of degradable polymers with regards to biointerfacing applications, implantable electronics. Are you also thinking about degradability in the environment in terms of kind of electronic waste? And if so, what would be the requirements of that particular application? Like compare, contrast, what would be the requirements, body versus environment?
Yeah, definitely thinking about that a lot now that I'm in Canada, because I think agriculture is pretty important here. So, I think a lot of the work that we've talked about today is at the interface of biology, so us and humans, and people are looking at the in vivo degradation of these materials. But I'm also very interested in thinking about how these degradable polymers will degrade in, let's say, in the ocean or in soil or in rainwater, like on a let's say you wanted to monitor how well a tree or some sort of agricultural plant is being farmed, right? So in some ways, the restrictions are less in the environment than in the body, because in the body, there's a lot of unknown, much more unknown and adverse side effects that could happen. And versus, like for instance, I work with carotenoids, and, people always say, just because it's in nature doesn't mean it's good for you. You could die from it. I'm like, totally get what you're saying. But, carotenoids and other types of compounds can exist in nature, and there's ways of degrading it in a way that is not harmful, right? So dialdehydes can exist outside of the body quite well, but inside the body, it's much more concerning because it can have all these additional reactive sites. So, I think that's one thing we're working on right now and trying to figure out. I think it's sort of scary, but I think I need to come up with figures of merit and follow sort of Jonathan in that area. It's always scary because I'm not used to leading in that way, but I think eventually that needs to be done as well. Mm-hmm.
Thanks, Helen.
Great. And as we're asking these more conversational questions, we'll try to pepper in some questions from the audience. We have one here for Sihong. Will the temperature affect the adhesion and the function of the polymer?
Yeah, that's a great question. So, for the function of the polymer, as long as you operate in a relatively mild temperature range, right? You do not go to over 200 degrees C, then it generally can, keep providing stable function. And for the user, that we are mostly interested in for interfacing with biological system, then you are operating in a, even smaller temperature window. So then regarding the adhesion property, if similarly, if you increase the temperature to too high, then it can also make it, overall less adhesive. It depends. It's I think it's overall, I should say it's a relatively complicated issue because adhesion depends on both the chemical interaction and the viscoelastic property of the matrix. And then we'll increase the temperature, you influence both. So the chemical aspects can become less stable, and but the polymer matrix will become more viscoelastic. So the we haven't from experiments perspective really studied the influence of a different temperature on the adhesion property, but I think that is a really important and a very interesting perspective. Thank you.
I'd like to ask Anna Maria. So a little bit going off of Sihong was mentioning the future need for multimodal devices. So I wanted to ask if you have been thinking about how you could create devices that have blebs from different cell types, or with regards to the AFM, it looked like it was a panel of T cells. Is it possible that you could do an immune profile where you have macrophages, T cells? Could you discuss a little bit about how you might envision that type of technology?
Yes, thank you so much, Alex, for this question. Yes, exactly. The ultimate goal is to develop a platform which can accommodate, which can multiplex, right? It can accommodate different types of cells. We like working with blebs to study the cell membrane because, of course, we don't have to have the sterility and all those intricate, conditions when you deal with actual cells. So that's why we are moving towards cell membrane events. So yes, we can. The idea is to add microfluidics where we can flow blebs from different cell types, have ways of even letting them interact together. We can do that also with live cells if we want to see at different levels interactions of live cells and tissue together by adding microfluidic technology, which coming back to what I was saying, really helps also what we've seen on standardizing also the measurements, because we have very strict dimensions where biology is restricted and in contact with the device. And so this really decouples a lot the complexity on our system when we have very strict dimensions and very strict surfaces of interaction between the material and the biological component. But another aspect is also, apart from multiplexing with different cells or different methods, it's also to use different methods to get a better understanding from one sample. So we can combine, for example, electrical, optical, mechanical properties. Even with AFM, we can do conductivity. Of course, this deviates from the scope of label-free, not label-free, portable in some cases. It's not as portable, but if we're talking about a diagnostic tool in the lab, it could be something really. So it really depends on the application. If we want something really portable, we stick into electronic methods of sensing. We add microfluidics that give us compartmentalization, hence different cells, different labs based on the application. But if we want to have something more information about something, we can have a device in the lab that can accommodate different types of measurements, so those types of multiplexing.
Great, thanks. I have a question for Helen. This one's actually from me, so I get to control the thing here, so I'll ask my questions. All right. So your system that you showed that showed both kind of doping on changing the acidity of the environment as well as degradation. I'd be wondering if you can kind of talk about the interplay. You had one slide where you talked about how it would dope first in certain conditions before degrading. Maybe you can expand on that, and then maybe. Is there any similarity in that doping process with systems like polyaniline that maybe there's a lot studied and a lot known about those systems?
Yeah, I think there's a lot of analogies that are there exactly with polyaniline. That's what we're studying right now, and we're also looking, at acids that don't degrade the imine bond but dope the system. I didn't show any of the work here, but we've doped it with iodine as well. And we're also looking, Sihong probably can predict this, but like self-doping systems as well, where you have some sort of charge where it can dope the polymer and turn it from a semiconductor to a conductor without actually cleaving the imine bond. And then you can add a different type of acid or something very strong to degrade the imine bond. We're also our next system is actually having it degradable, but not have the imine bond as well. So I think eventually we're moving in those areas, but step by step, trying to make improvements in there. And we're getting an evaporator soon, so we'll eventually understand the mobilities and stuff of these materials. But even before getting too caught up on that, actually, I think there's so much we need to learn about the design of the materials before spending all this time how to optimize one type of material, because we haven't reached that yet. We haven't even reached, we know it's not an optimal material because there's certain characteristics that we don't have. But yeah, I think there's a lot of analogies in there, and different types of acids can really, depending on the pKa of the system, as well as what type of acid, that we have the Lewis acid or not, all of that is what we're exploring right now.
Mm-hmm.
Great. I have a question for Sihong. And with regards to making implantable devices that are based on hydrogel materials and making them have tissue-like softness, I think one challenge is exactly how soft they are and the challenges that will be met with actually surgically implanting them. So maybe if you could talk a little bit about the surgical handling of your particular devices, and whether or not you think any special tools or new methods of surgical implantation will need to be developed for clinical translation.
Yeah, that's a really great question. So overall, for the surgical implantation, there are probably two general different cases. One is to implant on the surface of a certain organ or tissue. So then that's basically attachment. And then the other case is really inserting like into a tissue. For example, like inserting like a subcortical probe into the brain. So for those two different cases, the requirements and the surgical operation are also somewhat different. So for the first case, the attachment onto the surface. So from our experience in handling like hydrogel-based device, exactly as you mentioned, right? Sometimes too soft, not self-support itself. So then, in that case, we can use like a kind of a supporting frame that can enable like easier and more stable physical property of the device. So we can better handle it and then put it onto the surface. And then after it's already on there, right, in place, then we can remove the support and then further maybe apply more pressure to make it better conform. And then for the second case of inserting into the tissue, I think that poses a even bigger challenge for generally any type of a soft device, even flexible device. The bending stiffness is, may not be enough to overcome the resistance of the tissue. So for that, people has already like come up with different strategies using like carriers or like a temporarily stiffening the like the device through different type of mechanisms. So I think those combined with the new materials and the device designs probably can solve the challenges, the two very different requirements on the mechanical property. First is during the insertion period, and then after it gets implanted for the further long-term operation in the tissue.
Thank you.
Yeah, thank you for the question.
Great. So we're coming up on the last few minutes here, and I know we have a very large and diverse audience from all over the world that's curious about how you got to where you are. So I wanted to ask, the five of us here all work with similar materials, conjugated polymers, small molecules, specifically for biointerfacing and bioelectronics. So I wanted to ask you, when did you know that you wanted to spend your early career, at least, on pushing the limits of this materials class? Was it a specific paper, a specific experiment that convinced you that you wanted to kind of dive into this field? We can go in our normal order. Anna Maria?
So it's something that you know very well, but the audience might not know. When I was a PhD student, Jonathan was a postdoc researcher in France, and we were all the PhD students very inspired from his work, and it was really, let's say, a very positive. Not competition, but like looking at your dedication and how actually enthusiastic you were about those materials really inspired a lot of us, definitely. Then looking at actually in my applications, looking at the possibilities of those materials and actually how easy you could go, you could get some results, some really good results, really motivated me to push further. So starting from enzymatic-based sensing, Sihong mentioned some nice work that he also moved really forward with, like really conjugating the polymers with the enzyme, which was always something I had in mind, that this could be a really cool application, for example. And then working on with, for example, lipid bilayers, and seeing how a new technology platform can open so many different fields really motivated me to continue on that and really make me love organic electronic materials.
Great. Only a couple more minutes. So what are some highlights for you guys as why you're pushing this field?
Actually, when I was an undergraduate, I worked in an electrical engineering lab with CJ King Liu at Berkeley, and we were able to have extremely fine control in like CMOS fabrication. And even though I really liked that amount of control, it really showed me the limitations in trying to make bio devices using those technologies. So that's why I decided to sort of think about how to build the electronics bottom up rather than top down. And that sort of took me on a journey of chemistry. And here I am now working in more polymeric materials instead.
Sihong, real
Yeah.
quick.
Yeah, I'll take the last minute. So yeah, my interest in the technology that directly interface with our human body actually started from very long time ago when I started my PhD research. Although I was working on a slightly different topic, that's for harvesting energy from the human body. And then I also have very strong interest in electronics. So then this came up, sort of as a very natural combination of designing electronics that interface with our human body. And then the reason I'm also very interested, as similar with everyone here, to like a polymer-based electronics is mainly because I do see the great opportunity and the potential. So especially it allows us to like address the challenge that come across material and the device levels. So then you have a lot of things that you can like play around, you can tackle. So that's something I really enjoy, even up to date.
Great. Thank you so much. So that about wraps up our youth talk session. Let me just share here real quick. There we go. I wanted to share the e-certificates for the three of you for participating in the youth talks. Thank you to Anna Maria from Khalifa, Helen from University of Toronto, and Sihong Wang from University of Chicago. And I encourage everybody to log in on September 12th for the next volume of the youth talks on human iPSC technology. So thanks very much for joining us, and we'll see you next time.
Thank you.
Super, right on time.
不再是奇迹，不再是幻想，此刻真感觉全世界对我鼓掌。不必太在意身旁惊奇的目光，可以点点头，可以放声歌唱。我创造奇迹，我拥有梦想，我希望看见所有骄傲的脸庞。不再为曾经失败放弃或感伤，努力才是真的方向。I can， 没有什么可以阻挡心中无限的力量。I can， 你也能够像我一样飞越最高山岗。I can。没有什么可以阻挡心中无限的力量。I can， 你也能够像我一样飞越最高山岗。创造奇迹，我拥有梦想，我希望看见所有骄傲的脸庞，不再为曾经失败放弃或感伤，努力才是真的方向。I can， 没有什么可以阻挡心中无限的力量。I can。能够像我一样飞越最高山岗。I can， 没有什么可以阻挡心中无限的力量。
生日快乐！该吹蜡烛了，
来，我帮你。啊！